This is now part 3 of chapter 1. We have discussed some of the practices or the microbial world and microbes and human welfare, right? And we just finished learning about nomenclature and started looking into the history of microbiology. So in our last video, we ended with Jenner, Edward Jenner, for the smallpox vaccine, right? Hopefully this is still fresh in your mind. And now we are moving on to more history. Okay, so the idea at the time, which I explained also in the last video, was this theory called spontaneous generation. And this was widely accepted by all the scientific community and everybody at the time. They believed that living organisms just simply would appear from non-living material. And they saw this with their eyes, right? They would see toads suddenly appear, mice, snakes. So they figured these things must come from the soil. Flies from manure, and after a dead body starts to decay, maggots appear, right? And so from their experience, from what they witnessed with their eyes, this made sense. And so this was very strongly rooted in everybody's belief and understanding of the world. So when Francisco Redi challenged this idea, he set out to specifically disprove the idea of maggots spontaneously arising. And so he actually did 2 different experiments. In his first experiment, he had a sealed jar with meat that's decaying and an open jar with meat that's decaying. And in the sealed jar, no maggots ever appear, but in the open jar, you get the maggots. And he said that, there's some air in the sealed jar. How come you don't get the maggots? And the material itself, this non-living material that they believed was responsible, at least in part, to this spontaneous generation, it didn't happen. But the scientific community rejected his first experiment, saying the sealed jar didn't allow airflow, and that they believed that airflow was contained this vital force that would interact with the non-living material, and that's how things would form. So then he did a second experiment. He's like, fine, if you don't believe this, maybe you'll believe it this way. So he covered the jar, instead of it being sealed with a lid, he covered it with a net, kind of like cheesecloth, a fine net. And air is able to get through that. Okay. And then he had an open jar. And this actually was very clever and should have been enough to persuade the scientific community that, see, there is no vital force in the air. Things do not spontaneously just arise from non-living material. And it did really shake up the scientific community. But they weren't ready to let go of their spontaneous generation idea. So this started a debate that continued on for 200 years. And so we see another scientist at the time of 1745, his name's Needham. He found that microorganisms can be found in boiled broth once that broth is allowed to cool. So he was convinced that that was support for spontaneous generation, that the broth, being a non-living material, was able to give rise to more microorganisms after you kill them by boiling, that they just reappear after it cools off. Now, of course, you and I know That's because the microbes are in the air, and if you allow it to come into contact with the air, with the microbes in the air, then it will start to grow. So fortunately, there was someone around to say and point out this logic, and Spallanzani suggested that microbes come from the air in 1765, arguing against spontaneous generation. And he showed that you could have solutions that are sealed immediately after being boiled, and that they did not develop microbial growth. And so he's trying to demonstrate that microbes do indeed come from the air. If you leave broth exposed to air, it will lead to microbial growth. But if you seal it off, it won't. But again, this argument of you can't completely seal something, because how can you tell me that microbes are in the air, or if it's actually some mystical force in the air? Something in the air is leading to microbial growth. You're saying it's microbes, I'm saying it's a vital force. So this experiment's not valid to show which one of us is correct. And so then the debate continued on. And so then in 1858, Virchow challenged spontaneous generation with the concept of biogenesis, saying that living cells will only arise from other pre-existing living cells. However, he still didn't have scientific evidence to support this claim, just like Spallanzani did not have proof that microbes were in the air, right? So the debate still continued on until Louis Pasteur was able to demonstrate once and for all. And so Pasteur actually contributed a lot of major milestones for the field of microbiology, which it wasn't called microbiology yet, but we now look at this and see his major contributions to the field. So he first did some work, well, this wouldn't be his first work, but his first work notable that we're recording is his work with fermentation, because and I have a slide on the next page. We'll look into what these things are, but fermentation and pasteurization, and then he finally disproved spontaneous generation with a special set of experiments, usually referred to as the swan-neck flask experiments, and we'll look at that in detail. And all of this work was a major contribution to the germ theory. And the germ theory states that microbes can cause human disease. Not all disease is caused by a germ, but many diseases, such as infectious diseases, are caused by germs, which we call nowadays, we call these germs microbes. Okay, and so this was a major shift in thinking for hundreds of years. Well, for the vast majority of time, no one even knew about bacteria and microbes, right? Then for a couple hundred years, we've been discovering them, but we don't know what they are really. We don't know what their role is. And we're discovering this, and we're discovering the relationship between humans and microbes, and specifically pathogens. And so just like you've started to see this struggle with disproving spontaneous generation, there was also a struggle to get people to accept the idea of germs, that things that you cannot see are responsible for things you do see, like infection. Okay, so make sure you read about this in your textbook. All of these all of this history is laid out in your textbook. I may add a few little things here and there from other sources, but definitely read your textbook. It'll be very helpful for you to get this picture in your mind of what was happening over the years. Okay, and so then Pasteur went on to work on discovering what is the causative agent of rabies, and then from there he was able to make a vaccine. He also did this for anthrax, but not for people. It was to treat cattle, which is also very important. If you don't have cattle and if you don't have crops, how do you live? You wouldn't have any food to eat. So, taking care of the animals and keeping them safe from anthrax was huge. He also did this for a type of cholera, which was called chicken cholera. So, I already talked to you about Vibrio cholera, which is the human disease. And he worked on the chicken version, again, to maintain reliable food sources. Okay, so now we're ready to look a little bit more into fermentation and pasteurization, and then we'll look into the swan-neck flask experiments. Okay. Also, I forgot to tell you, Pasteur was French, as you probably know from his name, but he was an actual full-time scientist, which was new, right? In the past century that we were looking at, people had to have a primary job, and then the science was like a hobby. Now we're seeing in the 1800s people could actually be scientists as a profession, and he was a chemist first and then really got into microbiology. Okay, so fermentation and pasteurization. Some merchants asked him if he could figure out why sometimes their wine and beer would turn sour, and if he could figure out why, could he figure out a solution to stop this from happening. And it took him some time, but he started studying the microbes in these solutions, right? And he was able to study yeast, isolate yeast, and learn that yeast is able to utilize sugar, and their waste product is alcohol. And they do this without any air. So in a sealed container, anaerobic is what we call it, the absence of air. And so yeast will turn sugar to alcohol in the absence of air, and this is called fermentation. Okay. However, if you open up that solution of alcohol to the air, then bacteria will be able to turn the alcohol to vinegar, and it's these bacteria that are responsible for turning the wine and the beer sour. So these bacteria are they're in the air, and they may have already been present in the solution when the yeasts were doing their job. And as soon as they have air, they start to metabolize the alcohol, turn it to vinegar. So this was not only important in trying to solve this specific question of how to prevent wine and beer from turning sour, but it was also huge that somebody could finally definitively show that microbes are responsible for food spoilage, because this was not a concept that people had yet. This is a new concept. And so this was a huge step in the right direction to understanding microbes and disease, right? So if microbes can spoil food, what else can they do, kind of is the idea here. And so later on, it actually took him, I think, a couple of years, but Pasteur finally came up with the solution to the problem. So first he discovered fermentation, and then he discovered the bacteria that turn alcohol to vinegar. And then he came up with a way to stop this from happening, is to heat up the liquid enough to kill the majority of the bacteria, not so much as to ruin the quality of the fluid itself. And then this will give you a much longer amount of time before it spoils. And of course, now we know this process is called pasteurization, named after Pasteur, right? Um, and so he came up with this originally to treat wine and beer. This technique was then applied to milk because people could get things like tuberculosis, diphtheria, scarlet fever, salmonella, Yersinia pestis, which is the cause of invasion of the Black Plague, and listeria. All of these pathogens can come from the the cow. Not the cow is not infected, but it's on the skin of the cow, and it gets into the milk during the milking process, and then it's in a food source, the milk, and it's able to grow and get into a large enough number to cause infection if somebody were to drink it, or sometimes it's other ways that they would get sick from this. But we applied pasteurization to milk, and we were able to greatly reduce this infection from milk. Furthermore, the milk lasts longer before it goes sour. This was later then applied to other things like juices and such. So huge, changed our food production industry with this pasteurization technique. We have further since made more a lot of advances with pasteurization. We have different types, and we will actually get to look at that about midway through the course, maybe a little bit before that. And this is just your introduction to it today. Okay, so Pasteur's discoveries with food microbes led to the germ theory that microbes can be the causative agent of human disease. And then he went on to prove, so he would isolate the causative agent from a disease like rabies, right? And then he was able to go on further and make a vaccine for it. And so that was huge. That completely changed the practice of medicine. This idea that germs are in the air, germs are everywhere, and if they get into a food source, if they get into a wound, a surgical wound, right? Anywhere that they get into, it seems like they can grow and cause problems, whether that be to spoil the food or to cause an infection that makes you sick and kills you, right? So really changing the way that people think about the world around them. So how did he finally disprove spontaneous generation? Recall that this back and forth debate was, well, how do we know that it's microbes in the air or it's a vital force in the air that's responsible for the broth becoming contaminated with microbes after it cools off, after you boil it? So we look at some of the broth under the microscope before we do anything, and we see there are indeed microorganisms in this broth. Then we boil it, and we boil it for a while, and then we let it cool down, and we take a sample and we look at it under the microscope, and there's no microbes present. And so that was the first step, is like people had already done that, right? We already know there's microbes before you boil, and then there are not any microbes after you boil. But what we were seeing from previous experiments is that if you let it cool off, eventually it will have microbes in it as long as it's exposed to the air. But if you seal it, if you prevent it from being exposed to the air, then it will not spoil. And that was the whole debate. If you cut it off from the air, is it because there's microbes in the air, or is it because there's a vital force in the air? So Louis Pasteur had this challenge that he had to overcome, and it was brilliant. He came up with the swan shape flask. Here, my pointer. So see this shape of the flask, where during boiling, you can see steam is able to get out. Because you never boil a completely sealed container, right? It'll explode. And this shape here is the key to his success. It's why we were able to definitively, once and for all, say spontaneous generation is false. Okay, so you take a sample, you see there's no microbes present at all, but it's technically, this is open to the air, right? This is not sealed. Air can travel, and we can see that air can travel because we see the steam travel, right? Now, this is shown again after a long period of time. It has cooled, days have gone by, and it is still sterile. There are no microorganisms present. And this is why we were able to say spontaneous generation is not true. There's no vital force in the air. The air was able to get through. But this bend here, I don't know why they labeled it up here. It really should be here. This bend here would trap any dust and any microbes. So air was able to continue to flow through, but microbes and dust and all those things are not able to travel up after this bend. They couldn't travel up this tube and get into the broth, and that's why the broth stayed sterile. We even see that the broth stayed sterile for such a long time that it started, the broth itself started to evaporate. So after this evaporation, eventually they sealed it off so it would not completely evaporate. But you can see that was, months of leaving this flask open to the air. But microbes must be getting stuck down here because they never get into the broth. But the air certainly does get through. We know the air gets through because, well, we see evaporation here. And on that first slide, we saw the steam exiting out, right? So that's what it took to be able to show the difference between exposure to the air and exposure to microbes, right? So 200 years of scientists trying to prove spontaneous generation false. A neat story, right? Sometimes a little frustrating, but definitely very neat to see this. So, moving on. After all of Pasteur's work, after all the other scientists of his time and before him, when we get to 1838, we get the cell theory. And this says that all life is composed of one or more cells. Cells are indeed the most basic, smallest unit of life. There's nothing smaller than the cell that is alive. And then lastly, biogenesis, all cells arise from pre-existing cells. So sometimes the cell theory, these three parts of the cell theory, are attributed to Sheldon and Schwann. But really, they were reading everybody else's work and putting it all together. So they didn't come up with this off the top of their heads by any means, but they did get to make this official, right? And so here's a picture of one cell, and then it begins cell division. You can start to see it's expanding, it's doubling. Here you can see it's starting to divide, and here now you have two separate cells. And so this is just demonstrating that all cells arise from pre-existing cells. All of the work, not just Pasteur's, but everybody's work before that, kept demonstrating life comes from other life. It doesn't come from some magical, mystical force. So yeah, many scientists contributed. And really it was Koch and Van Leeuwenhoek who started that whole journey back in the 1600s. If they hadn't made their microscopes and made their observations, we wouldn't have even known about microbes. Okay? All right. So Lister, also learning all these things, decided to apply these concepts to surgery. And he was the first to come up with the idea of aseptic surgery where you disinfect your hands, you disinfect your surgical tools, and you disinfect the skin, the surgical wound. Yeah. So he applied the germ theory to medical procedures, saying that, yeah, germs, these invisible microbes, are causing infection, and we should take this into account with our medical procedures so that people don't have to die so frequently from medicine. He knew the midwife's work with childbirth fever, where we saw that women died much more frequently after childbirth when one of the doctors, who were men, would deliver the baby. But when the midwives, who were women, delivered babies, they had much better outcomes. And so he kind of put this together with the germ theory, realizing that doctors are touching sick patients, they're touching dead bodies to study them. And so their hands are probably covered in much more germs and much more infection-causing germs, composed of midwives who do cleaning and other things that are not going to have those same microbes on their hands. So that was huge. So he's building on all these other people's discoveries and putting them together. And then, of course, Pasteur's huge discoveries demonstrating that microbes are in the air, and that also we see that microbes can cause disease, such as what we saw with rabies, and that they can also spoil food. So we know that microbes are a big deal. The germ theory is real. And so Lister put all this together. He also happened to know of a chemical called phenol that could kill bacteria. And so that was a huge key in knowing what to do with all this information. I just learned about microbes being able to cause disease. What can I do about it? So he took his knowledge of phenol and decided to try it, to test it out. So he treated surgical wounds with phenol. He sterilized his surgical tools, and indeed his patients had much better outcomes. He had much fewer infections and fewer deaths using this practice of surgery. And so we call him sometimes we call him the father of modern medicine, because he began the practice of aseptic surgery. And that really did change medicine forever, for the better. Can you imagine being in that time where it was not common practice to wash your hands before you deal with another patient? Today, that's just common sense, right? Especially nowadays in this pandemic, everyone knows about disinfecting and washing your hands. But they were just figuring all this out for the first time. Okay, so Robert Koch, he is known for what we call Koch's postulates. A set of experimental steps that can definitively link a specific microbe to a specific disease. And this is great. We will later on talk about the shortcomings of this. It does have some limitations, but this was an excellent start in starting to learn about specific microbes and specific diseases. And so he was a German physician, doctor, okay, and he wanted to prove that microbes cause disease. He's working kind of at the same time as Pasteur. They both kind of independently did this work of identifying a particular microbe to a particular disease. So I know that can get a little confusing, but try to remember that, one person's in France and the other person's in Germany, and they're learning about all the same things and applying them. And so it's not that crazy that they did some of the same work. But this person, Robert Koch, did it in a different way, and in such a way that you could repeat these steps and apply it to other diseases and make a lot more quick progress. So while working to identify the agent of anthrax, he came up with his postulates, these steps to link a specific microbe to the cause of a particular disease. First, he had to establish a link between the microbe and the human disease. He later on, so he did this with anthrax, and then he later on did it with tuberculosis. As I said, Koch and Pasteur independently provided this definitive experimental evidence that anthrax, which is the bacteria Anthrax bacillus, was indeed responsible for the infection that we call anthrax, which is a respiratory infection that it can infect cattle, but it can infect humans as well. Okay, so as I said, Koch was able to identify the cause of anthrax, tuberculosis, and he also did for cholera, Vibrio cholera. He established experimental steps to identify disease, the specific microbe that causes that particular disease. And so let's look at those steps. First, the microorganism must be found in every organism that is suffering from that particular disease. And you should not find that microorganism in healthy organisms, so that you could say this organism is causing this particular disease. But that's not enough by itself. We need to prove it. So then we're going to isolate that organism from these diseased organisms, grow it into a pure culture, meaning only that one microbe and nothing else, and then put that pure culture into a healthy organism and see if they develop the same disease with the same symptoms that you started with in step 1. And then re-isolate. So if that organism does indeed become sick with the same disease, isolate the microorganism from that diseased organism, and then you'll know if it is still the exact same pathogen that you got from step 1. By the time you get to step 4, you know for sure, without a doubt, this is the microbe causing this disease. And this worked for a lot of things, like we saw anthrax, cholera, tuberculosis. It doesn't work for everything. There are microorganisms that can be fine and a part of your, what we call your microbiome. It's just on or in you. And under different circumstances, it can become infectious. And so that would not meet the requirement of step 1, right? And so there would be a lot of things eliminated. The other problem is that some people experience different symptoms. So it'd be really hard to say that, for everything to always fit his rules. His rules are super strict. They're beautiful in that you have no doubt. It's beyond, without a doubt, this microorganism is causing this disease. But it doesn't leave any room for all these other exceptions. So we used this for a while, and then we had to kind of modify this to take into account all these exceptions that we learned about later on. But this was an excellent start. He really got it started. Okay, Paul Ehrlic, he was the first one to come up with an antibiotic, but it's not a true antibiotic because antibiotics technically are natural. They're produced by an organism. So really the correct way to say it is that he was the first one to come up with a synthetic drug to treat a bacterial infection. So he had this idea, what if we could come up with some compound that could seek out the disease and kill the disease without harming the person. So syphilis was rampant at this time in the early 1900s, and he wanted to find a treatment for it. And so he tested hundreds of substances until he finally found one that seemed to kill this particular bacteria that causes syphilis. And so his drug, which was a sulfa compound named Salvarsan, and this was his idea of a magic bullet. And other people then took the same idea, and it worked for a few other drugs you'll see in your textbook. But again, this is a synthetic drug. It's just a chemical compound. Okay. John Snow, in the early 1800s to mid 1800s, he did work kind of on the other end. Instead of trying to treat a disease that people already have, trying to figure out how do these outbreaks happen and how can we prevent them. And so he's known as the father of epidemiology. He didn't do work that was only in the laboratory, but he did a lot of work in a community, learning the pattern in the community of disease, and put that with laboratory work to really get a better understanding of how diseases are spread. And he specifically worked with an epidemic of cholera, Vibrio cholera, in 1854 in London. And he suspected the source of this outbreak was from a water pump that was, he could see where people would dump their wastewater, which was, toilet water, essentially. Any waste. They dump it into this river, and it wasn't very far away from this particular water pump, and he made the connection. I said it's probably, the water pumped up the bad water with it and got people sick. And so he took samples, but all the samples he took were negative for bacterial contamination, which is kind of puzzling. But that just tells you that, it's very dependent on time. But would everyone believe that? No. So he needed to do more work to prove that it was this water pump. And so he created a map by talking to all the households in the area and all the households of people who had the cholera outbreak. And he created a map of every single person that had become infected and where they had gotten their water from. And he found the pattern of disease spread and distribution. And so by backtracking, he was able to show it is this water pump, and we should stop using it, or we should stop allowing people to dump their waste. It needs to be farther away so that it doesn't contaminate the drinking water from this water pump. So that was a huge discovery. If you want to see a little bit more about epidemiology, there's a really fun docudrama, right? It's not just a dry documentary. It's a reenactment. It's really fun. It's called Typhoid Mary, the Most Dangerous Woman in America. And it's the story of epidemiology in practice, finding out how this outbreak of typhoid fever was happening. So I encourage you to watch that when you have the time. Okay, we are almost done. Okay, Alexander Fleming is the first true antibiotic, and it was an accident. He was doing some work with Staphylococci bacteria, and he had some fungal contamination on his plates. And so this is the fungal contamination. These are the normal colonies that he was looking for. When he saw this contamination, he was going to just throw out the plates and start again with whatever he was doing. But then he noticed, well, I have a lot of colonies, up here, but the colonies down here are tiny, and then there's areas where there's no colonies, and he just through observation noticed that this colony is suppressing the bacteria from being able to grow near it. So then he started taking the penicillium fungus and working with that, and realizing that there are some bacteria that will not be able to survive in the presence of penicillium, and then that's where we're able to extract and develop a drug, which is the antibiotic penicillin. That took some time, but penicillin was available in the 1940s. And a lot of people think this is how we were able to win the war. So that's also something you can look into if this interests you. But again, this was accidental mold contamination. Anyone working with Petri plates knows that that's terrible. You should not be getting contamination like this. But thank goodness he didn't immediately throw it away. He looked at it, thought about it, and noticed this pattern. And that's how we were able to get penicillin. All right. So modern microbiology, you could study bacteria specifically, or fungus, mycology. Maybe you're interested in parasites like worms, helminths, things that can infect your intestine. Well, intestinal worms are just one type of helminth. There are many types. And then maybe you're specifically interested in viruses. And maybe you want to look into the field of virology, right? There's also the branch of immunology, which is huge, and applications of immunology are utilized in like every field these days. And then there's a field called bioinformatics, which is looking at the genetic material of microorganisms and learning about them by studying their DNA. There's a lot of modern technologies that have given us huge advances in the field and that are still being utilized. PCR, you'll learn about later on in our class. Recombinant DNA, I started to tell you a little bit about that. Genomic sequencing, which goes hand in hand with bioinformatics. They are separate, but bioinformatics only exists because we're able to do genome sequencing. And then we have huge advancements in imaging, different types of electron microscopy, different types of fluorescence microscopy, and then using antibodies in different types of tests, different types of lab tests. So that's connected to immunology, but we're able to apply it to all sorts of things. And then also we are trying to develop gene therapy as an actual practice that we can use in medicine to cure people with genetic diseases. So microbiology, we tend to only think about infectious disease, treating and preventing that. But now we're able to open up and start thinking about DNA. Because we've studied DNA in microbes, we're able to utilize them to treat ourselves. So that's really exciting. So what are the biggest current issues going on in microbiology? Well, antibiotic resistance is huge. It's a huge problem. Thousands of people die every year because they have an infection that we were not able to cure because none of the antibiotics work against it. Emerging diseases, which you're definitely familiar with now, as we've had this coronavirus outbreak. And then there's this huge issue with misinformation, people not understanding how vaccines work, and so they have these ideas that they're somehow dangerous, even though they are not dangerous. And so that makes it difficult because vaccines are one of the best inventions ever, and they're our best strategy to prevent disease. And if we cannot get people to utilize them, then it doesn't work, right? And then the other issue are crops. How do we have sustainable crops that are resistant to bugs, resistant to drought? We've learned some genetic engineering that works against that, but it's becoming outdated where we're getting resistance genes of bugs that can survive. And also we have a lot of public fear where people think that it's dangerous, or the food isn't as good for one reason or another. They are afraid of it because they don't understand it, and so they buy things that say non-GMO or whatnot. You don't need to worry. If you don't understand it, ask me about it, please, because I want you to have your vaccines and be safe. And I want you to be able to eat things and not be afraid that they're bad for you. And that is it for the lecture today. I have included practice questions for you. I'm going to go ahead and do one more video of the practice questions since this has already been 45 minutes. All right, take time, take a break, look it all over, read your book, and then try the practice questions. All right.